Hi. Do we have all the speakers in now? That's a very nebulous question, isn't it? If you're not here, you're not going to tell me. Right. So it's 11:15. We'll get started and keep everything on track. We don't anticipate any fire alarms going off. If they do, proceed outside. We'll all be directed to fire escapes and so on. Obviously, if you see that symbol, don't share anything on social media. When it comes to questions, ask you please to keep any questions nice and concise. Keep to the point because we only have a couple of minutes for those. So to start us off, Seth, how do you project up there?
It should just come straight up, I think.
Should it?
Yeah.
Yeah, cool. So, Robin Spenson, University of Gothenburg.
So, good morning, everyone. I hope you can hear me in the mic here. I hope you're not too tired from all that free wine yesterday. That was excellent. I'm going to talk about the roles of evenness and richness on community assembly and invasion. And more specifically, I'm going to focus on possible interactions between richness and evenness. So I come from Sweden, up north here, and on the northwest coast, we have a small island called Tjärnö, where we have a marine station, and this is where I spent about a decade working and then doing my PhD until one glorious day. The Swedish king dude gave me a bunch of money to go to Australia and do a postdoc, which I did. And I went to Monash Uni down in Melbourne, and I was working together with Dustin Marshall and his lab, the Marine Evolutionary Ecology group. And I was really fascinated and wanted to learn their method of making your own sessile community. So you start by taking a panel with plastic sheets here. You put it in the water. You just wait for stuff to settle and grow. And then you take it back into the lab and you identify a bunch of species that you want to work with. And then you simply cut them out from the plastic and you super glue them to a plate. And that actually works, which I didn't almost believe before I saw it myself. And then you put a bunch of plates on a panel, add some ropes and stuff, and here's what it looks like in the water. So maybe you can see the individual plates here and the panel. So I thought this whole method, the technique of making your own community would be a good idea to look at the diversity invasion paradox. So some studies find that the more native or the higher native diversity you have, the harder it is to invade. While other studies find that the higher native diversity you have, the higher exotic diversity you will have as well. So it doesn't really make sense, completely opposite patterns. So some people have looked at scale, and so this pattern is more small scale experiments, and this is larger scale or even landscape scale. And some people have looked at, this is usually manipulative experimentation, and this is more often observational studies. But I wanted to look at diversity, like the measures of diversity and the components of diversity. Could that response variable, how you measure it, could that be part of the pattern as well? So the two major components of diversity is richness, as I'm sure you all know. It's just the number of species, right? So two species here, 4 species here. The relative abundance, the relative distribution doesn't matter. Like the total abundance doesn't matter. It's just if the species are present or not. And then you have evenness, which is then the opposite. So it doesn't matter how many species you have. It's just a relative abundance. So this is maximum evenness. You can see the abundance is even, the relative abundance is even between 2 species and between 4 species. And that's the same value of evenness. And same for low evenness is when you have uneven relative abundance. And again, it doesn't matter how many species you have. So this means that for one level of richness, you can actually have several levels of evenness. So as you can see here, we have 4 species and 4 species, the same level of richness, but the relative distribution is even here and uneven here. So you get different levels of evenness for one level of richness. And the same is then true for evenness. You can have high evenness with 4 species or with 2 species or any number of species. And then if you combine these two, you get orthogonal levels of richness and evenness. So then you can look at a range of factors and you can ask if richness is high. Does it matter if evenness is low or high? And the same thing here. When evenness is low, does it matter how many species you have? So this is the design that we used to test for invasion. So you have these communities like this, and you add invaders from space or elsewhere, and then you see in which community they will thrive. Now these are the species that we chose to work with for this experiment. So we have 2 encrusting snails, 3 bryozoans, and a sponge. And this is the actual design that we used. So we had to cut 1,200 individuals or individual colonies of the same size for the same species at the same time and superglue them together to make communities. So this is panel design number 9. So you have 6 species here, 2 of each, so high evenness, or in fact max evenness. And then when you do all this, then you take your wheelbarrow and you bring the whole thing out into the field. Now this tiny speck is Marcelo, PhD student from Chile. Now field experiments in Australia is slightly different from field experiments in Sweden because everything is trying to kill you. This is the blue ring octopus that we commonly found on our panel, and I think even when I took this picture, it was just coming towards me. And if they bite you, your central nervous system shuts down and you basically die within 5 to 10 minutes. And then you have the black widow spider. This is Haley, who was an RA. She's now a PhD student. She found the redback, a type of the black widow spider, in her dive boot when we were out in the field. So in Sweden, we don't usually have to look out for venomous spiders and poisonous octopus. And then you also had a bunch of seals at the jetty, which I assume are poisonous and dangerous because they're in Australia. And after not dying in the field, this is what we found. So I'm not going to go through all this. I just want to show you the design. So you have richness and evenness, the interaction, and then panel is a random factor, so all the interactions with that. And then we looked at the whole community for assembly purposes, and we looked at the invading species, and we had 2 samplings. And this is what we found. So for the most common invader was a hydrid called Laumidea. We found an interaction between richness and evenness, and it seemed they preferred, or we showed the highest cover of the species when richness was low and when evenness was low. Like that's the communities where they thrived, grew the most. And then we looked at number of invasive species settling in our communities. And we found that only richness mattered, not evenness. And we found the difference was between the high richness treatment and the medium treatment. It was not different from the low, which we found a bit surprising. And then if you look at the total cover of all invasive species, we saw the same pattern, but it was only a trend. Oh, sorry, it's only a trend. So you would see more when evenness is low and when richness is low. So interaction here. And then when we looked at the total cover of all species, total abundance in the communities, we saw a completely different pattern. There was an interaction between richness and evenness, but it turns out the abundance of the whole community, you get the highest cover, the highest abundance when richness is high, but evenness is low. And then for total species number, like the total amount of species coming into the community, we again saw that richness mattered, but not evenness. And there was a difference here between the high and medium treatments of richness, but no interaction with evenness. So, in conclusion, richness and evenness interactively determine cover of the most abundant invader and trend for total invader abundance. And there was also an interaction, but a different pattern for the total cover of all species. And then we also looked at multivariate statistics, the composition of the community, and that was as well. We saw an interaction with richness and evenness. And richness alone determined the number of invading species, as well as the total number of species. So just in conclusion, we found that the levels of richness and evenness matter for both community assembly and biological invasions. So I just want to thank some few people. Swedish King Dude giving me money. Science Councils for also giving me funding. And Dustin and the whole MEG lab with the RAs and PhD students. And that was it for me. Thank you for your attention.
Thanks, Robin. Do we have any questions for Robin?
What are you using as your measure of evenness?
Pelouse evenness, which I think is the most common measure.
Thank you.
Do you have any other questions?
Yeah.
What do the communities compete for? Is it only the space?
Yes, commonly they say that space is the major resource they compete for. But they actually have also been shown recently to compete for oxygen and for food, for instance. So they compete for a lot of resources besides space, I think. Yeah.
With regard to experimental design, I wonder whether you put out your plates
Yeah.
at one location in the field, so like the occurrence of the invading species may differ on a larger geographical scale. Did you put it out on one spot, or did you kind of put it out far apart?
For this experiment, we just wanted to look for interactions between richness and evenness. So we chose one spot where we knew there would be lots of invasive species, because we didn't want to gamble with a design like that. It takes forever. So that's what we did.
So the replication was within the
Yes, one side only. Yeah, that's true.
Got time for another question? Okay, I just got a quick one. Did you look at occurrence of different taxa within the invasive communities themselves? So propensity of different taxa to invade these communities you have?
Like which invasive species?
Yeah. Which ones are more likely to invade given the different setups?
No, but that's a good idea actually. Yeah, I'm gonna do that now. Yeah.
Credit. No,
Yeah,
okay.
yeah.
Okay, thanks, Robin.
Yeah. Thank you.
Round of applause. So next up we have Ron Bassar of the University of Oxford.
Thanks, Anthony. So what I want to talk to you guys today about is predicting coexistence among species that with structured life histories, continuously variable structured life histories. A long-standing question in ecology and evolution is, do structured life cycles impede or facilitate species coexistence? And what I'm showing you pictures of here are different types of structured or complex life histories that are based off of discrete traits. So for example, with frogs, you have tadpoles, froglets, adults. And with things like butterflies, you have going from chrysalises to caterpillars to adult populations. And this is typically what most research on structured populations and coexistence has sort of focused on. And the theory suggests using these, with these types of discrete life cycles that can lead to a variety of different types of coexistence outcomes. And coexistence is generally thought to be possible when at least one species shifts its niche and each species is a better competitor in one of the life stages. And the stage, the species where they shift their niche, they're also the better competitor in that stage. Otherwise, you get predictions such as competitive exclusion or a variety of different types of priority effects. But not all populations, particularly in mammals and many animals and many plants, are structured by these discrete stages. And quite often, populations are structured more based on size, which can be related to age, but also just size in and of itself. And so the question that I'm going to try and address today using a modeling framework is to talk about how do life cycles structured by continuous traits, using size as an example, impede or facilitate coexistence. And there's been a fair amount of theoretical work done on this using what you might think of as size-structured community resource models, wherein you have two or more species competing for one or two different resources, and then the model's coexistence emerges from the interactions between different sizes of individuals in those populations. And these are great models, and they're very useful for thinking about how coexistence emerges from these types of very intricate interactions. But oftentimes, because they're consumer resource models, they're oftentimes envision them competing over one or two different resources, but unfortunately, a lot of species are not specialists, and so it's not unclear how well these models translate. And then also competition involves more than just food oftentimes. But probably most importantly is the predictions from these models are often very difficult to take out into the field and test. They're very difficult to parameterize for any given system. Another type of approach which is a much older sort of approach, is to use what I like to call competitive interaction models. And these are typical types of models where you measure the competitive effect of individual species on other species, such as in the Lotka-Volterra competition equations. Now, these types of equations can very easily be made into having discrete traits, right, where you have different interactive effects of different size or different ages or stages. The approach that I'd like to describe to you guys today is using a newer type of modeling technique based on integral projection models. And you can think of integral projection models as basically continuous size versions of matrix population models. And this is the sort of standard, this is the equation that's often used. And this is, so you're projecting some distribution of population sizes from one time point to another. And those population distributions change based off of something called the transition kernel, which describes growth, survival, and reproduction of different sizes of individuals. Now, if you're going to talk about competition, the first place you need to start thinking about this is in a single population model, single species model, and of course you need to talk about density dependence. For any given vital rate, such as growth, survival, and reproduction, density dependence most oftentimes in these integral projection models is included by having a negative effect of total population size on some vital rate. This is just the vital rate. The vital rates also change as a function of size independently of total population size. In a paper I published last year in Ecology Letters, I wanted to first sort of ask the question, what are the assumptions using these types of models? And basically the assumption is that competition, when you model competition as a function of total population density, you're assuming that competition is what's called symmetric. In other words, every individual has the same effect on every other individual. So in this model, I decided that we can do much better than that, so we can actually measure, we can model the density dependence using capturing asymmetries in competition. And asymmetries, of course, are where different individuals based on their traits have different effects. And so this is just a starting point to sort of point out that the way you can do that is using what are called interaction surfaces, in very much the same way that you would do with discrete size structured models, except now you're talking about a continuous surface. Of course, if we're going to talk about coexistence, we're going to talk about two different species, and we need to talk about broader types of interactions among individuals. And this is what a trait-mediated two-species model of one of the any given vital rate in the population would look like. Again, you still have the density independent component and how the vital rate changes as a function of size of species I. But then you also have the interspecific interactions denoted by one term here and the interspecific interactions denoted here. And you have, in each one of these, you have an alpha II for the intraspecific and alpha ij for interspecific interactions. Oop. And so now the challenge is to think about, okay, how do we begin to model what types of alpha surfaces, interaction surfaces do we need to have in order to get coexistence, both with intraspecific and interspecific. And of course, the big thing that oftentimes is what allows for coexistence is the niche differentiation, right? And so oftentimes individual different species may occupy different niches, but also not only do different species occupy different niches, but oftentimes those niches change through ontogeny. And so this is an example from Arctic cod from the Beaufort Sea, where small individuals are feeding mostly on, it looks like calanoid amphipods, but then their niche changes as they go through different stages of ontogeny. And so the key thing that we want to be able to capture is these niche changes both within species as they develop and between species and how much their niches overlap through time. And the way to do that, as I mentioned before, is through these these interaction surfaces. And I just want to point out here that these alpha surfaces are made up of two components. The first one is this competitive asymmetry part here, which is described by this equation. And this is basically just saying that competitive ability increases with body size, and it may do so differently between the species. And then this one here is just a general difference between the species. The key part about the niche differentiation component is this Gaussian function over here. And what this means is this along the x-axis here, it says the difference in size between any competitor and the focal individual size. If you're the same size, which is denoted by 0, you have an interaction surface that should be equal to 1. And as the size differences change, then those competitive interactions decline. Basically, all you need to do is to multiply these two together for any given size, and you get this function over here. And so this is now an alpha surface that takes not only into account the niche differentiation, but also how competitive symmetries change with size as well. And you do that for all focal sizes of focal individuals and all different sizes of competitors. You can get a variety of different types of interaction surfaces. And these are just sort of a single parameterization of these models just to show you the variety of types of surfaces you can get. The purpley sort of areas are sizes that interact the most strongly, whereas in the white areas are size combinations that don't interact. And so you can get, for any single type of parameterization, you can get intraspecific interactions that change with size, but where the competitive asymmetries are symmetric. So these guys over here are not really interacting with these guys here. And you can get a variety of different types within these. And so the question is, how does the shape then of these different interaction surfaces influence the coexistence? So I'll use a single parameterization of this model just as a sort of demonstrative tool to see what kind of overlap there is with previous types of models. And the parameterization is based off of a system that I do a lot of work in, which is the Trinidadian guppy. And what I'm going to talk to you about, the parameterization is based off of the two different species having the same life history. And this is what's commonly done in a lot of theoretical treatments of coexistence. And the reason to do that is just to be able to control the general life history, so the age schedules of birth and death, and then just be able to look at how the differences in the interaction surface influence coexistence. And I'll present to you just really quickly 3 different ecological scenarios. The first is that where individuals of both species shift their niches with increasing body size, and there's no size-based competitive asymmetries. The second is the same, except that now that we're going to have competitive ability of one species increases with body size and the other doesn't. And the final one is what's predicted to happen when the competitive ability of one species increases with body size and competitive ability of another species decreases with body size. And this final one is the scenario that's most often pointed to when in discrete models where you were able to get coexistence. So the first result, this is where coexistence, where there's no differences in size-based asymmetries, but both species can shift their niches to different degrees. So down along here, you have, this is the amount of overlap that you have among different sizes for species 1 and species 2. And so there's complete overlap among between small individuals and large individuals. So in other words, small and large individuals occupy the same niche, all the way up to another part of the spectrum where the small and large individuals are completely separated in their niches. And under this scenario, whichever species, this diagonal line here represents neutral coexistence, where basically the species are identical. And what this says is that whichever species is able to shift their niche more than the other one is the one that will competitively exclude the other species. Under the second scenario is where we start to get patterns of coexistence, and that's basically where one species 1, exhibits competitive asymmetries with increasing body size. And so when there's complete overlap amongst all individuals of species 1 and a range of different overlap of species 2, then we get coexistence emerging as a prediction in this region here. Finally, the final prediction is that when we have increasing asymmetries in competition with species 1 and decreasing with species 2, not only do we get coexistence in about the same region here, but we also start to get priority effects out in this region here. And the priority effect is whichever species is in the habitat first is the one that will be able to competitively exclude the other. So finally, just to conclude that these predictions are fairly similar to what you get in the context of discrete models, but these types of models here allow you, if you're working with size-structured organisms that are structured in continuous space, such as body size, you can actually take these models out into the field and try and parameterize them to ask questions about any species of interest. You can also do sensitivity analyses, which I haven't shown you, and there's a number of different extensions, I think, to these models that I hope will be utilized soon. And with that, just want to say thanks, and thank you all for attending. I'm probably out of time, aren't I?
You're all right, man. We got time for a couple of questions.
Cool.
Yeah. Any questions for Ron? For Ron? Sorry, Ron? No? Oh.
Yeah. Jason.
Have you explored the impacts of changing this Gaussian niche separation? So if you, for example, have maybe longer tails.
Yeah, so you can, all I've changed is like looking at the mean, like how the mean changes as a function of body size. But you can change, one extension would be to change the niche breadth, and look how that sort of influences coexistence as well. There's a number of different extensions that I think like that you could do into it. You can have abrupt changes as well. It doesn't have to be continuous. There's all kinds of things like that. Yeah, there's another question over here. Yeah.
I was just wondering if you, I know this isn't hit the sort of topic you haven't talked directly, but I was wondering if you had any thoughts about how the overall availability of resources might affect the dynamics between two species. If you had a resource.
Yeah, so these models are not, there's no resources in these models. They're two species competitor models. So I think that's a very hard thing to do and answer within the context of these models. Because basically what you would do is to change those betas. Those betas oftentimes become more towards zero if you have an increase in resource. But I don't know that these models would capture what you get in like consumer resource models where you have increasing resources and competitive exclusion based on that. I don't know. Yeah.
Okay. We'll have to move on now.
Yeah,
That's all right.
yeah.
Thanks, Ron. So next up, we have Mikael Pontap of the University of Zurich. Yes, thank you.
Okay, thank you very much for the introduction. Thank you for this opportunity to present some work that I've done together with Owen Petchy at the University of Zurich. So before I go into the details, a very general aim and interest introduction. I'm currently working on understanding richness patterns, trait distribution patterns, and also phylogenetic patterns. And to do that, I use ecoevolutionary simulation models. This is, of course, a general interest in community ecology, and it's of particular interest for inference techniques that are aimed to infer unknown processes from patterns that we see in nature. And given that we succeed in this task of inferring processes from pattern, that of course feeds back to our more general understanding. This talk will be mainly on this first more general point, but I will frame our results in relation to inference techniques. So, oops, that's not good. Lost a bit of the screen here. Oh well. So community ecology and inference techniques are often based on one common inference technique is based on inferring habitat filtering and competition from community clustering. And that was introduced by Webb. And this technique kind of assumes a species pool of a regional species pool of species with different niches, different traits maybe. And that species pool then assembled into local communities. Yes, could you? Perfect. Would be great if you can actually see the whole talk, not only the half. Better. Thank you very much. No. Oh well. Okay. So a species pool. From that species pool, a local community is assembled through either competition or the other extreme would be habitat filtering. That then leads to an overdispersed community, assuming that similar species are excluded through competitive exclusion. The other extreme would be a clustered community containing species with very similar traits that kind of dictates those species' survival in the abiotic environment. These ideas then kind of underlie the inference techniques that uses overdispersion or clustering to go back and infer the major processes that dictate the assembly. These techniques has been used quite extensively, but they have also been criticized for excluding processes. Criticized, for example, for excluding trophic interactions. And with that being said, that of course is somewhat limiting, especially if similar processes or different processes like competition and predation gives rise to similar patterns. With this in mind, our aim here is to kind of look at the predatory effects on the prey community structure. We ask two questions. First, how does predation affect prey community clustering? And second, we ask whether these kind of classical web inference techniques actually work in the presence of predators. Based on literature, we make two predictions. First of all, predators can punctuate the prey community trait distribution through invasions and subsequent extinctions, and thus cause lower prey community clustering. And kind of the flip side of that prediction would then be predation at extreme ends of the competitive community trait, the prey community trait distribution, would kind of narrow that trait distribution down through extinctions and then increase community clustering. To answer our questions, we focus on, or we use a 3 trophic level community model. We assume a regional species pool, so again, following kind of the web assumption. We assume a regional species pool of J prey, so J prey and K predators. And we also assume that these prey and predator has some kind of trait, V or Z. Furthermore, we define a basic resource distribution or resource availability. We define J resources in that same trait space, and here that trait will be denoted by U. So we model this as a classical Lotka-Volterra predator-prey system, but we expand on that model to make it trait-based, and thus assume that trait matching dictates resource utilization, very much like in the previous talk. And from this, it follows that similar consumers compete more than less similar ones. And you should have seen, up here, there would be these the trait-based interactions being modeled by a function expanding on that Lotka-Volterra model. More specifically, we assume, so here those functions are actually visible, so A-pred and A-con dictates the interactions, so the ecological interactions, and we model that explicitly through Gaussian functions, also like similar to the previous talk, where, for example, the interaction between predator and prey is dictated by the traits of the two focal interacting species, which is maximum when they are overlapping, and then that kind of interaction falls off according to this parameter sigma pred, and it falls off symmetrically as a Gaussian function. And the same interaction between the basic resource and the prey is modeled in a similar way. We will focus on parts of this model to answer our questions and test our predictions, and those will be the sigma con, which is the niche width of the prey. That parameter kind of dictates the overall competition in a community. And we will also focus on the niche width of the predator and the efficiency of the predator. And we will focus on habitat filtering, which here is defined by the overlap in the resource trait space, overlapping with the prey available species pool. If that overlap is large, we will have low habitat filtering. If it's small, we will have high habitat filtering. I need to rush a bit because this was... okay, so we model two specific scenarios. We use the model, we simulate the assembly of communities. Two specific scenarios. One kind of reference point scenario would be just having predators. So we invade predators with different degrees of competition and different habitat filtering. This will give rise to, so again, the extremes would be an over-dispersed community, and on the other extreme, a clustered community. We use these communities as reference points when we then introduce predators with different predator niche width and efficiency. And we look at the effect on community clustering. The assembly process involves 3 technical steps. I won't have time to go into detail here, but the first thing we do is we set up the resource, the basic resource distribution. We introduce a prey species. We compute the equilibrium population sizes. We do that through numerical analysis. This is actually the simulations of where we have 2 predators, a number of prey, and basic resources. When we have that, we can define or we can compute invasion fitness throughout trait space, both for predators here in red and prey, the gray. So we get these fitness landscapes. And then we pick species with positive invasion fitness. We introduce them. We do actual invasion trials. And that's done through, again, computing equilibrium population size. And we iterate over these 3 steps over and over again until we have a fully assembled community, and the output will then be these trait distributions. Those trait distributions we analyze, we compute mean trait distance, MTD, and mean nearest trait distance, MNTD, 2 matrices that are often used to quantify community clustering by comparing those values to a randomly assembled community, which will then allow us to quantify community clustering. So to the results, the reference communities, starting with the reference communities having only prey in there, we see that the richness, prey richness, yes, increases with prey niche width, decreasing prey niche width, and it also increases with lower habitat filtering. So note here that habitat filtering decreases along the x-axis. Community clustering decreases along that same parameter space. Okay, picking a number of reference communities and introducing predators, we then see that predator evasions lead to extinctions. No surprise there. The extinctions increase with predator efficiency. The number of predators here shown by the different columns, and also with predator niche width here denoted by the colors. Colors down here. We also said, so that was not very surprisingly, but more surprisingly, we see a general decrease in community clustering as predator invade. We see that here. There's a decrease in MTD-based clustering with predator efficiency and number of predators invading, and also the niche width. And these results are quite robust over the parameter space, and they are robust for the different MTD, MNT values. So to my final conclusions and things here, sorry for the I was a bit disruptive in the beginning. Okay, thanks. Okay, so the reason for this decrease, so that's the interesting part. We want to understand the mechanisms that drive these patterns. And we can see that in this figure where we show the basic resource distribution, the prey distribution, the prey fitness landscape, and the predator fitness landscape when 2 predators has invaded the system. So the reason for the decreased prey community clustering is because predators tend to invade evenly across trait space, and that can be seen in this fitness landscape where these peaks are quite evenly distributed. That leads to extinctions and in the prey and decreased clustering. So we confirm our first prediction. We do, however, see that predators tend not to invade the peripheral parts of the trait distributions. And that is due to low invasion fitness in the peripheral parts, basically. So predators increasing community cluster is unlikely, and we don't see it in our simulations. And finally, we saw an unexpected effect, and that's these evenly distributed secondary extinctions in prey. So we can see that here, where there has been extinctions in prey, even though they are not directly affected by the predator. And that's due to invasions of predators changing the abundances of the prey, and these kind of secondary competitive exclusions. And those exclusions are also tend to be evenly distributed. So the conclusions: predators can give similar patterns as competition, which is interesting. Velen has suggested it. Here we show it explicitly for these two processes. This, of course, implies that classical inference techniques that look at these rather simple statistical relationships cannot tease apart predation from competition, which is a concern. And we thus also conclude that more process-based and process-oriented inference techniques that takes multiple sources of information into account is needed. And this is something we're currently working on, we and, of course, others as well. Thank you for listening. Very sorry for the layout of the talk. If you're interested in more of the details, please go to our recently published paper in Proceedings B, and I want to thank Owen, and I want to thank all of you for listening. Thank you. Yes, there's no...
Thanks, okay. No, unfortunately, we don't have time for questions at the moment. Sorry about the technical mix-up there. Sorry.
Yeah. The laser is this one.
Okay. How do you start a...
You go to full screen mode. So here in view. Yeah, and full screen.
Okay.
Thank you.
Sorry, you have another technical hitch. It's a PDF presentation.
I guess you go to... If you say full screen,
Yeah. What's the shortcut of F? Function.
F11, maybe? If you press F11, it might just be the screen. No, sorry.
If you go to the row.
Yeah, and again.
Yeah.
Oh,
Hey.
Control L, it is. There we go.
Sorry about that. So, Ingrid Olivares, University of Zurich.
Thanks. So I'm going to talk about, this is a work in progress. It's actually the first chapter of my PhD thesis that I have been developing with my supervisor, Michael Kessler, and a postdoc in our institute, Dirk Karger. So first of all, I'm going to give you some context about why I'm doing this research. I work for a project called Popcorn, which stands for Using Population Genomics, Phylogenetics, and Community Ecology to Understand Radiations in Neotropical Mountains. in this project, it's a collaborative project among three universities in Switzerland, and we tackled this question on what causes and limits the radiation of species from three perspectives. So some of us work on evolution, and they work on phylogenetics. We work on the distribution and interactions of species at different spatial scales, and some other of us work on the genomic basis of diversification. I particularly work here in the ecological component, and I look at the distributions and interactions of species at the local scales of communities and assemblages. So we wonder how could we, from the community ecology, contribute to the understanding of species radiations. And some of you might know this paper from Peter Linder that is called Plant Species Radiations, Where, When, and Why, in which he shows the figure for what a radiation is. And he explains that it's when there is an initial rise in the diversity that lasts for a few million of years, and then this decreases or slows down. And this kind of pattern is known as saturation or bounded diversity. And we wonder, what does saturation mean? So we started reviewing papers, and we noticed that it's used in different contexts, from community ecology to evolutionary biology. And we set up this aim to review the concept and methods used to test for it. So far, we have read more than 100 papers. And these papers have a big range of research. So one of the classics is this one of Terborg and Fabborg, in which they looked at the saturation of bird communities in the West Indies. This has also been studied using invasive species and different kinds of organisms. And there are other there is an important group of papers that don't use the term saturation, but are very important for this debate, because the methods they use and they are central for studying saturation, but they don't use the term. They use like bounded diversity or limits. And we are also focused on those. So one of the first persons that actually used saturation in an ecological context was Charles Elton in this book of 1933, The Ecology of Animals. And here he asked how many animals can live together, how many animals of a similar kind can live together in a community. And he concluded that except in very unfavorable conditions for life, the number of different kinds of animals that can live together in an area of uniform type rapidly reaches a saturation point, which we can just illustrate with this basic figure. So we see that there is a saturation of the animal species after going through a specific number of samples. Later on, MacArthur and Wilson used the same term in a very different context, in the context of the theory of island biogeography. And what they explained is that saturation or equilibrium is the point where the immigration and emigration rates collapse or cross. So, and if we can imagine that if we sample the number of species through time, we will kind of see a similar pattern. So they introduced time into the equation. And so we wanted to unify this concept of saturation, and because there are many studies that say, that saturation can be demonstrated, and there are many that are against it. And we figured that it is because most of the studies don't specify saturation in relation to what. And it is very simple, so we say that saturation is a continuity of pattern that must be defined in relation to where, when, and of whom. And although this concept can be very obvious or can look very intuitive, it hasn't been put into words in any paper. And we just want to provide a definition in very general terms. Saturation is the spatial or temporal stability of a species richness of a group of organisms under certain environmental conditions within a certain time period. So we have one principle, but we have several methods that we can use to test for it. We have this pattern of saturation or curvilinearity of a species richness, but this can be studied, this can be studied using time-related variables in two scales, ecological and evolutionary time scales, in which the drivers are different, or we can study it using a relative maximum. And this relative maximum is usually a regional pool that can be of taxonomic, functional, or phylogenetic nature. And while we have been doing this categorization, we have been also extracting all the mathematical expressions, so because the idea is to have like a guide so that people can use when they are interested in testing a particular concept of saturation. So here I'm gonna go a bit more into detail. These will be the categories for testing saturation in ecological time, which can be studied using species accumulation curves. This is the more basic concept, like the one from Elton, in which we will look at a number of replicates and we will see what is the upper limit in the number of species that we can find. And comparing like different localities, we will find different levels of saturation. The other one that goes into the ecological time category is this one of succession, in this one we will compare the immigration and emigration rates and trying to find what is the equilibrium point. But this has to be studied in a longer ecological time scale. Then the same can be tested in a geological time framework, and in this case we will look at the same patterns but using speciation and extinction. So this is in a more longer temporal scale. And then we can also have this relative maximum as the related variable. And in this case, we have local species richness versus a species pool. Here, the basic concept states that saturation occurs when the relationship between local richness and regional richness reaches an asymptote. So if the local richness continues to increase with increasing regional richness, then we will have unsaturated communities. And this can also be studied using functional groups. So instead of comparing against taxonomic regional richness, we can use functional richness or functional pools. Or we can also study this independently from the spatial scales. Just by asking how is the relationship between taxonomic categories and functional groups. And there is an example from this study from Knope and collaborators in which they look at how many ecological modes are occupied in different taxonomic categories through different time periods. Finally, there are some fields that we would like to explore, and is using chaos theory to study saturation, because we think that one of the main problems in demonstrating and testing for saturation is the use of linear dynamics, because it is a dynamic process. So using linear dynamics might not be very straightforward. And for those that don't know, chaos theory studies the behavior of dynamical systems that are highly sensitive to initial conditions, a response popularly referred to as the butterfly effect. This butterfly effect, or chaos theory, became famous when Lorentz set up these experiments to predict meteorological outputs. So he found out that having two curves that have a very minimal difference in their starting point can lead to very different outputs. And this is actually something that can happen with saturation. Having very little difference in the initial conditions can result in very different outputs. So we think that this using nonlinear dynamics might be useful for studying saturation, and this is another application of chaos theory, which is studying the movement of a pendulum. And so if it doesn't, of course, show like a linear dynamics, but it shows this other kind of pattern between two states. And this is something that we also think that might be found while studying saturation. And I would like to thank the Swiss National Science Foundation for funds for our project and all people in Popcorn project from these 3 universities in Switzerland. And thanks to you for your attention. So I guess we don't have.
Yeah, well, we got time for one very quick question if anyone has anything. No? Okay, we'll move straight on then if that's all right. Where do I find this thing now? No, never mind.
I guess we will have the same problem, yes.
Ah,
So I prepared the slides in 16:9 ratio, but well, I think most of the graphs will be fine.
yeah, there's nothing I can do from
Yeah,
here.
it's fine.
Sorry.
I will just start.
So we got Robin Alter from the University of Zurich.
So thanks for your interest, and so over the next few minutes I will talk about how the network structure or dispersal between habitats, connectivity between habitats is influencing biological communities. And I will present results on amphipod communities in particular. So it has been notion for a long time that connectivity and dispersal between catchments are influencing biological communities. So it's not just the habitat properties that are influencing biological communities, but also how they are interconnected. And if you remember the first talk of this session, there it was more about invasiveness and how community structure is influencing invasiveness. And here I'm more focusing on how connectivity of habitats is influencing invasibility of habitats. And if you look at this picture, you might wonder, or you think of a community in this little stream here and another one here. So even though having very comparable environments, they probably show very different communities just based by the different connectivity of this and this habitat by this lake that is somehow dampening connectivity. So I will focus on aquatic ecosystems, and this nice alpine landscape probably looks very different from the point of an aquatic organism. So just the dispersal or connectivity for an aquatic organism looks like this tree or so-called dendritic structure. That is a very common and universal attribute to aquatic or river ecosystems. And it's not just looking very nicely, it's also, this tree structure also has influence on, for example, species richness. So we expect just by the difference in connectivity to have certain richness patterns emerging. Also differences between species turnover within the network. And this is not just...
I think we can get the wide aspect if you can. Sorry.
Yeah, sure.
Sorry about that.
Great. Thank you. Thanks. So these are not just theoretical expectations. It has been shown, for example, in fish communities. Here is the Mississippi River in the United States, and here you can see that from headwaters downstream to the outlet side of the Mississippi, fish species richness is increasing. There are models also on this topic, so just from the different connectivity in these networks, you can see that more central nodes within the network show a higher diversity compared to more headwater sites or sites at edge. And this is really a difference to, for example, these more grassland type of networks. But up to now, all these results and models have been just looking at native species, and as you know that with connectivity there is going to be invasibility and non-native species, and we are now interested in what is about non-native species within these networks. As I'm speaking in front of ecologists, I don't have to explain much about non-native species, but particular in freshwater ecosystems, they pose a big and sometimes detrimental effects. And here you can see a picture of, for example, the Asian clam in Lake Constance. So you can just grab these non-native species, and they can have really a huge influence. And we are using amphipods as a model system to study the influence of the network structure on native and non-native communities. And amphipods are very common in these ecosystems within Europe, and they have a very important ecosystem functioning, or they represent an important ecosystem function as being a bridging part between leaf litter and hydrotrophic organisms. And luckily, or more to say interestingly for us, we have native and non-native species, and for the rest of the talk I will denote native species in black and invasive or non-native species in red. So the questions we wanted to answer is, how is amphipod diversity structured in these river networks, or influenced by the network structure, by the connectivity? What patterns can we see, and are there differences between native and non-native species? And can properties or properties of the network explain some of these differences that we might see? So we conducted our study in Switzerland, in the central part of Europe, and over the last few years we collected a huge database about Swiss amphipods with more than 4,000 records and about 170,000 individuals identified to species level. That is like the phonistic data that we rely on, and for the spatial data we looked for, for this talk I will focus on the Rhine catchment. So these are more headwater sites, and then the Rhine is flowing downstream through some lakes. Here is Basel, so here is the outlet site to the North Sea. And we constructed a adjacency matrix for this huge catchment. Plotting first results, we saw the expected pattern of increased diversity downstream and lower diversity of amphipods in headwater sites. Things are getting more interesting when looking at how these species are distributed along the network. So on the left-hand side there are headwater sites, and then moving downstream along the network you end up in the outlet sites. And when plotting native species along the network, you can clearly see that they are quickly increasing as expected. More species or higher species richness in more downstream sites. But we can also see that there is, like as mentioned in the previous talk, there is somehow a saturation. So we seldom or rarely see more than 4 native species out of 7 possible native species. On contrary, and very interesting, when plotting non-native species, one can see that in headwater sites we don't see any invasive non-native species, and then a quick increase in non-native species being even more abundant or present with more species than native species at the most downstream sites. Combined together, we can see the expected pattern of increasing diversity along the network. When looking at beta diversity or species turnover within the network and comparing sites along the topological distance, so along the river network and not as aerial distance. One can see also, as expected within native species, a distance decay. So the further apart two sites within the network, the more dissimilar the amphipod community. But again, when plotting non-native species, we can see a different pattern with no distance decay of diversity. So this suggests that there is either that non-native species are not dispersal limited, or that there is some sort, or the combination of with habitat filtering. And with these plots, one cannot see the difference. So one of these universal properties of river networks is that they are directed. And comparing, even though this distance along the network is the same topological distance than this distance, it's very, so the distance would be the same, but comparing two headwater sites is really a difference than comparing a headwater site to the outlet site. And to look at this, we we wanted to retain the hierarchical structure of the network on the left-hand side, and we compared it to the dissimilarity of communities. So first results, it's really work in progress. You can clearly see that there is some sort of clustering going on, and this suggests that communities assembly within these networks is happening in some modular way. So the network really has an influence on community assembly of these amphipods. So to the key findings, we found these distinct patterns of amphipod richness and amphipod communities along this network. We found that non-native species are mostly restricted to larger, more connected streams, whereas natives are less abundant in downstream sites, but that headwater sites present some sort of refugium. And these results were robust across a big range of spatial grain sizes. And with these key findings, I would like to thank my lab, the Ultimate Lab at University of Zurich and ERWAC, and all the people that helped and the funding agencies. And I would like to thank you for listening.
Good time. So we have time for a couple of questions, if anyone has any. You just answered everything, didn't you?
No. Cool.
No questions? Okay.
Let's see if it works.
Don't hold your breath. Another one. Okay.
I think so, yeah.
16:9. Yeah. Sorry.
Yeah.
So next up we have Lalix Calastronier of the University of Liverpool. Did I pronounce that correctly?
Yeah, Henry, yeah, that's right.
Huh?
Yeah, that's right. Good afternoon, everyone. Thank you for coming. Yeah, my name is Lalis, and I'm a PhD student at the University of Rio in Brazil. And I did part of my PhD also in the University of Liverpool. And me and my supervisors, Joe Creed and Matt Spencer, we are interested in the factors that drive beta diversity patterns in a rockshore system in a bay in southern state of Rio in Brazil. So first of all, we have to define what we are taking as beta diversity. There's been some debate in the literature about these concepts. And I'm using for beta diversity what we think is the original proposal of Whittaker, which is the extent of change of community composition or degree of community differentiation in relation to a complex gradient of environment. So for me, what I'm interested in is trying to understand what environmental factors and spatial factors are responsible for community composition. And when I mean composition, I mean presence and absence in rockshore community. And I'm thinking this in a framework of meta-community, that is a set of sample points or a set of sites that are interconnected by the dispersal of multiple species. So people have thought about this a lot already, and many processes are thought to be responsible for community composition variation. So some of them are, for example, some of these processes are biotic interactions, stochasticity, source and sink dynamics, but I'm not talking, although they are interesting and I'm interested in them in my PhD, I'm not talking about them today. I'm talking about how environmental filtering is capable of, if it is capable of explaining some of this variation. So our study site, as I told you, is in southern state of Rio. The city of Rio is right there. So we have a bay there in southern state that is called Ilha Grande Bay, which means big island because it obviously has a big island on it. So we put 42 sample sites in it around across the whole bay and around the big island. And that's it. I put some photos here so you believe it's a true place and not just a map. So in each one of these sample sites, we collected information on 31 environmental descriptors. You can see from the photos there that the places change a lot in, for example, hydrodynamics and exposure to waves, etc. So we took all the information on environmental descriptors as we could think of. And we also collected data for 6 taxonomic groups. I represented those by the commonest species for each one of them. They are macroalgae, cnidarians, echinoderms, mollusks, crustaceans, and reef fish. So what we did was basically scuba diving along 100-meter transects, making visual sensors for hard bottom organisms, that is cnidarians, echinoderms, and macroalgae, and also reef fish. And for the soft bottom organisms, we took cores from sand and identified them later. So with the environmental variables, they were too much, so we did a principal components and took the 7 first axes out of them and used them as environmental variables to like a regression model. For spatial explanatory variables, we used a standard method called principal coordinates of neighbor matrices, the PCNMs. I'm going to explain them a little bit better further down the line. But we use those 2 sets of variables, the environmental ones and the spatial ones, to run GLMs for multivariate data using the package MVBond in R. And we decided to go with a AIC-based model selection to search for the best models. And for assessing explanatory power of each one of the models, we use the D value, which is analogous to the R-square statistic for linear models. So it has a similar interpretation. So our results in total, we found 768 species. Most of them were mollusks, as you can see here. And the distribution in the bay looks a bit like this. So the bigger the bubble, the richer it is. And the colors are a heat map, where the hot colors show places with more unique species more rare to find in the bay. So you can see from this that the west side of the bay concentrates most of the rich and unique places, let's say, like unique compositions, whereas the central part of the bay holds more commonly seen species, and also around the main island you got also some rich and unique places as well. And when we took those environmental and spatial variables for each one of the taxonomic groups, we found that, well, first of all, what strikes is that most of the variation in composition is unexplained, and that holds for all the taxonomic groups, and actually for crustaceans and mollusks, we could not find any environmental or spatial descriptor capable of explaining this variation. but for the other groups, we had, for example, for Nigerians and fish, we have a good explanatory power for the spatial modeling. Whereas for algae, for example, we had a better explanation with the environmental variable. The environmental variables. When I say environmental variables, I mean I mean, most of the time, the first principal component was the one that got selected most often. And it's mostly related to depth variables in general. So you have, in the bay, you have like, again, in the west side of it, you have the shallower waters. And as you would expect it, you have also shallow waters around the coastline, whereas the central and around the island, you have the deepest waters. So this is coincident with the pattern that we saw for the species compositions, in which you had the most uncommon places in places that are the shallower. So you got some of the explanation for this by depth. Now, yeah, it's definitely not working. So I told you that I would explain a little bit more about the spatial description of these models. They get a bit complicated, so I'm trying to make it easier. So what you want from a spatial model is that it is capable of giving you like the spatial arrangements of the sites that you got, and you got all available spatial arrangements that you can get from the set of points, and then you select from those ones that are relevant to your system. So the PCNMs, what they do is, first of all, you build a matrix of distances between all your points. So you have here for point 1, 2, etc. So, and then you find the minimum distance capable of linking all the sites together without leaving any site unconnected. So, for example, you can think that for the bay, this is a map of the bay, and all the sites here interconnected. And you can think of a link of a length of 100 kilometers, for example. It would connect all the sites because all the sites are within the range of 100 kilometers. But that's not what you want. You want the minimum distance capable of doing the same thing. And for this system in specific, it's around 10 kilometers. So with a link of a length of 10 kilometers, you are capable of uniting every single point. And from this, you can break it down into separate little arrangements and the whole thing. So by the end of it, you got a set of multi-scale description of your system. And you get all the distances that you get here. You hold only the distances that are equal or less than this threshold that you found. And all other distance, you just put them an arbitrary large number. So you got a truncated distance matrix by the end of it. And with a PCOA, you got values that are not, the scores are not particularly important per se, but what they give you is something like this, in which they give you the relationship among points like this. So black is positive, white is negative, and the bigger the bubble, the further it is from zero, either negative or positive. So what you get here is basically showing what things are close together and if they are similar together and how you can like see the general pattern out of it. So when we see this for the bay, you see that, for example, these ones were the ones that got most often selected. And you can see that they describe stuff looking alike, like in big scale separation. So you can get the spatial variables that got selected are like big scale stuff. I mean, this one is the biggest one. It separates the bay into two big groups of points. And this one is a little bit more intricate, and so on and so forth. So in summary, what we found was that rockshore communities in the bay can be arranged in 3 major diversity areas. Sometimes it works, sometimes not. Oh. Yep. Uh, Community composition is partially driven by depth-related variables and broad-scale spatial patterns. And most importantly, I guess, is most of species composition was unexplained by any of those, which is interesting. I mean, it could be a caveat of, I know, a local sample point or something. But since it is a common trait among other systems in literature that most of variation could not be explained, it could be a real trait of meta communities in the sense that composition in meta community might be selected or driven by other processes other than environmental filtering, for example, or spatial organization. So the next few questions we want to answer is exactly this. Is it a case that you have other drivers for community composition? Or is it the case that maybe environmental filtering is not that important for composition, but might drive abundance and differences in these systems and etc.? And explore also other technical questions, like is it possible to make variation partitioning in this GLM framework in which you can get how much of overlap between your environmental explanation and spatial explanation you can get from the system. So I think this is it. Thank you very much. I'd like to thank those people and those guys here for the money, and thank you. Yeah, I think sometimes it works.
So we're back on time nicely. So, and you're just leaving. No. Does anyone have any questions on that? So we got a couple of minutes. We're back on time after the hiccups. No. So I was just wondering what stochastic drivers you're going to be looking at.
Well, stochastic.
I know it's outside your presentation, but
Yeah, it's stochastic means stuff that you can't
oh
control.
no, I know, but I was wondering how you're going to be.
For example, you can look at how likely it is to get the same size of these communities, but without any driver in specific. So you can run like no models and see if what you observe is basically what you get when you have no drivers at all. So that's what stochastic is meant for.
Right. Okay. Cool.
Okay.
How do I find this? Oh, there it is. There you go. I've gone to search tools now.
Can okay.
There's a tab thing open. I don't even know where the. Oh, there it is. Right. Cancel that. No. Right. Let's try that.
Maybe now.
Yeah. Nope. Joy. See, the full, yeah, yeah.
The last This one, yeah. Here's
Yeah, let's do it to the next slide. Let's hear.
okay.
Sorry? Oh.
Thank you.
Sorry.
Okay. So I will present to you a study realized with my PhD, Michael Legal, and it was financed by with some partners, and it's on determinants of Rodonate communities, spatial assemblages along a blue way in Normandy, in France. No, okay. Don't want to put the next slide. Sorry.
I just use the
The arrow's not working.
No. And no.
I bet all the other rooms are fine.
I have to click here. Yes, okay. So, thank you. So in introduction, this study is based on species assemblages, and we know that species are not distributed in the same way, and it's and whatever the taxa on our landscape context. And if you see a site, you have a potential total species pool, but actually the species in your site are defined by the geographical species pool and the habitat that you have on your site. And also, all species are determined by the ecological species pool. This is the interaction between your species. And finally, you have the actual species pool. And we wanted to respond to three very important questions for management. And it was how species come and disappear from communities, and how communities respond to disturbances through time, and how ecological continuities are possible. And in France, we work on a very useful tool that is Greenway and Blueway, and it's based on networks of terrestrial and aquatic continuums to promote biodiversity and in very fragmented landscape. Considering only Blueway, because we work on Rodonats, mainly active water are often studied, and we don't know why, but ponds seems to be less interesting. And yet ponds are regularly described as potential relays in this Blueway, but we have no scientific proof for this. In very disturbed landscape or fragmented landscape, ponds can be considered as suitable patches in a fragmented matrix, and they are frequently described as a hotspot of biodiversity for plants, insects, and a lot of taxa. However, ponds are also subject to many threats from human activities, such as nutrient loading and pollution, agricultural intensification, and we have no information on the effects of this pollution and disturbance of ponds. We work on Rodonats, and it's a very interesting group to study ponds for few reasons. They disperse sometimes over large distances according to species, but also according to the surrounding landscape. Their life cycle makes them dependent of aquatic and terrestrial habitats. Therefore, they can be very good indicators of the ecological quality of the habitats. And Hodonat are divided into two suborders with damselflies, the smaller one, and dragonflies, the larger ones, with different dispersal capacity and different compartments for territoriality, clutch activity during the journey. So we will work for this study on two hypotheses. The first one is that Hodonata communities are determined by the biotic and abiotic characteristic of ponds, but also their surrounding landscape and their geographical location in relation with other ponds. And the second hypothesis is very important because if you have two ponds with good habitats, the important thing is that the individual can disperse between your ponds. And the second hypothesis was that pond networks allow gene flow at the regional scale and support exchanges by dispersal processes. To respond to the first hypothesis, we used 31 ponds situating along two rivers in the Seine-Maritime. It's a French administrative region. You have the Jordan River and you have the Seine rivers. And we work on the different ponds situated in different contexts. We have field ponds, forest ponds, and urban ponds. For the sampling, very quickly, we used five temporal sampling occasions to account for the phenology of all species. And we studied different characteristics of ponds, the size, the distance to the closest pond, the temporality of the pond, the suspended matter content in the water, freshwater macroinvertebrates that can be considered as prey or predator according to the species for larvae, and the vegetation in three belts around the pond, in the center of the pond, on the bank, and at 5 meters from the pond. The result. First, we use Montel correlogram to show the similarity between our assemblages and according the distance between ponds. And for the two suborder, we found that we had more similar assemblages when the distance was lower than 6 kilometers. Then we compare the similarity of the assemblages for the two suborder, damselfly and dragonfly, between the different contexts of ponds. And we compared urban with urban, forest with forest, forest with urban, field with field, urban with field, and forest with field. And we used a Kruskal and Wallis test. And for the two suborder, we only found a significant difference between forest pond and field ponds. So it's very interesting because you have dissimilar assemblages between forest pond and field pond, and in urban context, you have ponds with a mix of the species of the two other contexts. We also realized a Montel test to compare matrix of similarities between damselfly or dragonfly and the characteristic of ponds. And we also used partial Montel test. And we have more similar composition of damselfly when we have more similar community plants assemblages. We also have two ponds with more similar freshwater macroinvertebrates have more similar damselfly assemblages, but we have also a positive effect of the distance. We have the same result for the level of mineral matter, and finally for damselfly, and finally we have a correlation between the similarity of freshwater macroinvertebrates and dragonfly assemblages, and considering the distance. So assemblages of damselfly and dragonfly are not influenced in the same way by pond's characteristics. We also see the species richness, and for dragonfly, we have a positive effect of the pond area, of tall aquatic grasses abundance and prey abundance. And we had a positive effect of prey abundance for damselfly. So, species richness of damselfly and dragonflies are not influenced by the same pond's characteristics. To respond to our second hypothesis, we used 32 ponds to study the gene flow of one species of damselfly, Ischnura elegans. And this hypothesis was based on the fact that we have two watersheds here, and we have no hydrographic component between the two watersheds. We used 35 tarsi of male and 25 tarsi of female on each pond, and we used 8 microsatellite markers. And for the result, we identified two spatial genetic clusters in green and in red here, but we have no relationship with the spatial distribution of our pond. So the work here presented here is in progress, but we have identified a genetic structure. Then we show the kinship to see the isolation by the distance, and we showed that there is an isolation by distance between individuals within the studied pond networks. And finally, we have not found a significant relationship between the FST, the genetic coefficient of differentiation, and the distance. So, the pond located between the watershed of the Seine River and the Durand River allows the passage of the individuals. It's a very important thing. And we have probably a stepping stone model. To conclude, at the landscape scale, we have a switch of the species between our context, and especially between forest context and a field context. And in terms of management, it's very important information. Yes. Ponds, they contribute to the studied greenway and blueway, and whole ponds do not play a similar role in maintaining odonet communities and landscape connectivity. So, for management, ponds must not be considered as isolated elements, and the success of a pond network can therefore depend on the distance that you have between your ponds. And dragonfly and damselfly must not be considered in the same way when you want to manage hodonets. Finally, at pond scale, if you want to manage, local characteristics can be determinant to shape hodonet communities. For habitat selection, we showed that we have not the same variable that impact damselfly and dragonfly. And also for the reproduction, we have an effect of, for example, an effect of vegetation communities on damselfly, probably because all species of damselfly present here use endophytic oviposition. And we have an importance of the abundance of aquatic graminoid for dragonfly, probably because, and the size, of ponds, probably because most of species of dragonfly are very territorial species. I would like to thank all my collaborators, and especially Lucie, Michael, Pierre-Loup, and Audrey that collaborate with me on this project. Thank you.
Sorry. Do we have any questions for that talk? Yes?
That's really interesting, thanks very much. At the end, in your last slide, a lot of the sort of habitat selection criteria, you could see them being related to sort of the successional dynamics of the different ponds across the landscape. Say, if some of these ponds were constructed 10 years ago, 20 years ago, do you think there's any sort of influence of the sort of successional stage at which the pond is at on its sort of utilisation conceptualization by going for those individual dams and ponds?
Yes, we have probably a selection, but I have not present here, but we have also information on larvae. And we have found in which we have found haddock, but no larvae. Yes, yes, but I have not time to present all my results. And probably we have some haddock that we have detected, but it's not for reproduction, only for farm food. And we have not today the succession of pond stage, but probably it would be interesting to respond to this question, but only on larvae, not on haddock.
Yeah, of course.
But here I have not the information to respond precisely to your question.
Okay, so we have to move on to the last one now?
Mhm.
Okay. The last presentation is by Dustin Ranglek of the University of Nebraska.
Thank you. It's great to be here to talk about, this was some of the work that I did during my time as a PhD student at Utah State University, and it was published in the Journal of Applied Ecology and ended up winning the Southwood Prize Award for the best paper by an early career researcher, which is really why I'm here to speak to you today. Otherwise, I don't know that there'd be many people who'd be too interested in learning about bison and cattle in Utah and some of the interactions between them. So I'd like to acknowledge my co-authors, Dr. Susan Durham and Dr. Johan Dutoit, on this project as well. So oftentimes when we're managing natural systems, there's an attempt to integrate local ecological knowledge into the management schemes to try to capture some of that information, whether this is from indigenous peoples in the area or just long-term resource users, like ranchers in the western United States who've been on that landscape for many generations. And so oftentimes this looks just like a number of people gathering around a map and talking about different places they've seen or done things. But ultimately we still need to make sure that science is an important part of this management plan, as I'll demonstrate. When it comes to wildlife and livestock conflict, there's really a worldwide negative view towards native large herbivores. And a lot of that extends from concerns over the transmission of diseases, as well as the potential for competition between the species. And really, it's led to the near eradication of many wildlife species as they've been completely removed from the landscapes and then replaced with cattle or other livestock. And that's exactly what happened with bison. Historical estimates of bison in North America were between 50 and 60 million bison, and that shrunk down to just nearly 100 individuals at one point. Currently, there are about 500,000 bison in North America, though only 20,000 of those are what we consider to be in conservation herds. The rest are raised in commercial livestock production, which you can go to the store and buy bison meat. Of the conservation herds, most of them are intensively managed on fragmented landscapes that are very small and usually fenced. And many are experiencing some level of integration with cattle genes as well, as bison and cattle have been historically interbred in an attempt to create a super grazer. And really the main thing that's limiting large-scale restoration of bison is this perceived competition. And I really like to show, let's see, where's the laser button? Oops. I like to show this painting kind of depicting manifest destiny and the westward expansion across the plains. This button. You see here there's some bison up in the top corner running off into the distance, and for most of their native range, bison have never returned after they have been replaced by agricultural interests. So if bison and cattle are competing, we can kind of make two main predictions. The first would be that if bison are significantly reducing forage availability for cattle, we would expect that bison visitation to any particular site would be a predictor of grass biomass depletion. And second, if bison are the main wildlife competitor with cattle for grazing resources, we would expect that bison and cattle would be our two predominant grass consumers. To examine this, I worked in the Henry Mountains. This is located in southern Utah, here designated in red. This is about 125,000 hectares. That's area that's actually used by the bison. As a point of reference, Salt Lake City is right here, so it's about a 5-hour drive down to this area. It's extremely isolated, remote mountain range. We were 2 hours from pavement as we were working down there. And it's primarily public land managed by the Bureau of Land Management. About 99% of this is public. In the low elevations, it's a very sparsely vegetated desert grassland and shrubland community. This picture makes it look like there's actually more vegetation than there is. It's mostly bare ground. But as you move up, you get into pinyon juniper and mixed conifer communities, and eventually subalpine grassland and aspen as well. Bison were introduced into this area in the 1940s. It's kind of right on the edge of what would have been their historical range, and there's some debate about whether they were there historically or not. But this is a free-ranging bison herd that is genetically pure, disease-free, and hunted annually, managed by the Utah Division of Wildlife Resources, to be at about 325 adults post-hunt. Really, this should be like the crown jewel of bison conservation that everyone talks about instead of Yellowstone National Park, because we don't have any disease issues here. It's just kind of this slower population size that's keeping it from being a more well-known place. But the reason we chose it is this is one of the very few places where you have bison and cattle commingling on a landscape with no attempt to keep them separate. Generally, it's, bison stay in this pasture in their fence, cattle can be over here in their fence, and no mixing. But on the Henry Mountains, we do get a lot of commingling. But there are other grazers on this landscape, particularly lagomorphs, that we don't often think of, both black-tailed jackrabbits and desert cottontail rabbits. And once you get out of your truck and start walking around, you notice a large number of lagomorph fecal pellets on the ground, so we thought that they may be having an important contribution to the overall grazing community here. So our goals were to measure the rancher perceptions of this livestock conflict in the Henry Mountains, and then determine the relative impacts of both bison, cattle, and lagomorphs out there, and then kind of use the perceptions versus the reality to hopefully guide some management going forward. So we mailed out a questionnaire to the 21 ranchers who graze cattle in the area that's used by bison, and asked them to identify what the major issues are when it comes to having bison and cattle on the landscape. Are they concerned about competition for forage, for water, because this is a desert community, for space, disease, and so on. We asked them who they felt the main wildlife competitors were, if it was bison, lagomorphs, or mule deer, which are also present in the area. And finally, 2 questions about predator control. First, if they thought it should be done, and second, which species they felt benefit from that, as there's a very large amount of coyote removal that takes place in this area, government-sponsored. We received 12 responses from those ranchers, so 57%, which is lower than we would have liked. But all of the main largest producers in the area did respond. So we're representing 71% of the cattle that are permitted to graze in this area. And really, competition for forage was their biggest and truly only concern. And it was consistently rated as a high concern in every season and every habitat type. For wildlife competitors, again, a very consistent trend that they're worried about bison and bison alone. And really, when it comes to lagomorphs, it wasn't even on their radar that rabbits might be having any sort of an impact. And all felt that coyotes should be controlled and that it benefited both wild and domestic species. One rancher even wrote that he felt that it benefited all wildlife. I wonder what the coyotes would feel about that, but that's another story. So we selected this one particular grazing allotment. This is an area that's used as winter range for cattle, so the cattle are released in November and taken off in April, because this was an area that the bison used very heavily in the summertime, and so there's a real intense conflict. The ranchers in that area feel that by the time they're allowed to release their cattle, there already isn't any forage available for them because the bison have been there all summer. And we placed 20 grazing exclosure sites at random, following a few conditions. They had to be a certain distance from a road, etc., so we could actually set them up. At each of these 20 sites, we placed one 8x8 exclosure that would exclude bison and cattle, but allow lagomorphs to still have access. A second that would exclude bison, cattle, and lagomorphs, and then an open reference plot that would allow all grazers. So 40 exclosures and 20 reference plots in total. These were set up in October 2011, just before the cattle were released into the area, and then we returned a year later, clipped the standing crop of vegetation, and then separated it into grass forb, shrub, and cactus, and dried and weighed it for analysis. I'm just going to talk about the grass component now, because we didn't find any significant differences in our other vegetation types. So this is what it looks like on the ground. In this particular site, site 19, I show you because the ranchers repeatedly took me to this particular spot and said, this is a problem. This is where the bison are having a major impact. And so our partial exclosure, you can see it's just a cattle panel with T-posts. The rabbits can easily get in and out, and we did document that they were doing so. And then the full exclosure is the same design. This is just 10 meters away from it. And it has chicken wire that goes 2 feet up the side and 2 feet out along the ground to prevent burrowing. So this is what it looked like when we set up, and when we returned a year later, this is what we found. So if you do the comparison of the inside of the partial exclosure to the outside, that's essentially your large herbivore impacts. Where if you compare the full exclosure to the partial exclosure on the inside, that is your small herbivore impact, or your lagomorphs. In this particular picture at this site, it seems like our large herbivores are having very little impact, but the lagomorphs are having a very strong impact. But of course, this is science. We look at all the sites and not just cherry pick. So to revisit our predictions, the first was that bison visitation would be a predictor of grass biomass depletion. We had 44 GPS collars on bison in this area at the time, and so each of these black dots was representing one GPS location that was collected during the year that we were monitoring this area. You see that we did a good job in capturing, in placing our plots in areas that are actually used by bison, and capturing a lot of variation in use. So here's that number 19 I just showed you. The bison do use that area very heavily, where down here some of these sites were only visited once or twice. To quantify those differences, we used a relative density index. So we created a square kilometer buffer around each site, counted the number of locations that were found within that buffer, and then divided that by the number of locations you would expect in any given square kilometer of that habitat type. This was log transformed for analysis, so essentially any RDIs greater than 0 would indicate higher than expected bison use, and RDIs less than 0 would be lower than expected. So this is what we would predict. As bison use is increasing on our x-axis, we should be seeing an increase in grass biomass depletion. But this is what we actually found. If anything, this is a negative trend, but it's not significant in any way. So there's something else that's really driving the patterns of grass biomass depletion in this system. So we can see that bison visitation is not a good predictor. Our second prediction was that bison and cattle would be the two predominant grass consumers. So here we used a one-way randomized block design ANOVA to control for differences among the sites. And what we found was significant differences among the reference, partial, and full exclosure types. Essentially, the large herbivores are accounting for almost 14 grams per square meter of grass consumption, where our lagomorphs are accounting for 7.2 grams per square meter. Both of those are significant. But the question is, how much of it is bison and how much of it is cattle? In this area, there's over a 90% dietary overlap between bison and cattle, and because they're the same body size, they consume roughly the same amount of forage, so they're considered to be equal in terms of their consumption. So we're able to use animal days to determine the relative impacts of each. Basically, the number of individuals that were found in the area times the number of days they spent there. So for the bison, we used our GPS data to come up with about 21,000 bison days. For the cattle, we used data provided to us by the ranchers in the area to be about 82,000 cattle days. So when we break down that large herbivore component based on animal days, what we find is that cattle are consuming 11 grams per square meter, the lagomorphs is still that 7.2 from before, and bison only 2.9. So on a percentage basis, you can see that lagomorphs are actually having more than twice the impact of bison in this particular area. So we can see that bison and cattle are not the two predominant grass consumers, but it's actually cattle and lagomorphs. So thinking back to where we started, when we're integrating local ecological knowledge, in this case, the rancher's perceptions, we need to do it very carefully because it can be based on misunderstandings of the complexities of the system, as we see potentially with the impact that coyotes would have on bringing lagomorph populations down where they all felt that that was important. It might not be matched to current conditions, or they could be reporting their perceptions with the bias intentionally to support their political needs. They know that if they complain about rabbits, nothing will happen, but if they complain about bison, that can be a big issue. So management that's based purely on perceptions is unlikely to lead to satisfactory outcomes. So science really should be providing the main direction here. And just as a point of discussion, it's possible that we're seeing a trophic cascade from predator removal. Utah spends $1.35 million annually on predator control, and the Henry Mountains are targeted quite heavily as part of that. So if we have an unnaturally low coyote population, that would lead to a high lagomorph abundance and thus their major impact on the grass. But overall, I think that this provides a lot of hope for bison restoration. If you're going to find competition between bison and cattle anywhere, it would be here, where the resources are so limiting. And so the fact that we're finding very limited competition really gives me hope that we can see broad-scale bison restoration in the future. Thank you.
Any questions?
What did the ranchers think of yours?
That's always the first question, so I can be prepared. No, it's great. I developed a really good working relationship with the ranchers while I was there, and I tried to be very transparent and communicate with them as much as possible. So when I first presented this data to them, I got to the slide with the pie chart, and they said, "Wait a minute, you're telling me that rabbits are having twice the impact as bison?" And I said, "Yes." And there was this murmur that went through the room, and I was expecting to hear, "Oh, this guy's full of shit." But what they were saying was, "We need to stop shooting coyotes." So I thought that was really great that they made that jump immediately without having to be prompted. But then the next thought was, or we could just shoot jackrabbits as well, which I think is their current preferred alternative. So.
Time for one more.
It's kind of just that Old West mindset of, dominating nature. Utah spends all that money as part of the Mule Deer Protection Act that the state legislature passed. In theory, it's meant to protect mule deer fawns from predation so that the hunters can have more mule deer to shoot. But really, it's been found to be highly ineffective and a complete waste of money. But yet, it made it so that they have money to hire government contractors to go out and kill coyotes, as well as provide a bounty for public citizens who kill coyotes. So you get $50 for every coyote you kill. So there's a lot of people who really enjoy going out and doing that. And so they're very happy to have that financial incentive to go along just with the recreational activity, if you can call it that. So it's just kind of this vestigial idea, the Old West of dominating nature, I think. So.
Draw it to a close there. So thank you to all the speakers. Sorry for the technical hiccups, many of them. And yeah, draw this session to a close. So thank you very much and enjoy your lunch.
And initially we were doing that because we were concerned that maybe they wouldn't see the case that you're saying